Hi everyone, thanks for coming back to the last session of the afternoon. I'm Jeremy Yoder from the University of Minnesota, and I'm going to talk to you today about some fairly new work that I'm doing looking at how we go about identifying locally adapted loci in genetic and genomic data sets. So local adaptation to variation in things like altitude, water availability and temperature, and the presence of other interacting organisms is really a topic of perpetual interest in evolutionary ecology. We find that local adaptation is extremely common when we go looking in natural populations, and we're interested in it because we think of it as often the first step in ecological adaptive speciation processes. But also, local adaptation can provide natural experiments in adaptation to changing environments. And sort of traditional transplant-based experiments to diagnose local adaptation have been very good at finding it and understanding how it's currently going on. But to understand how it arises and what's led up to local adaptation that we currently observe, we need genetic data. And it's recently, since I started my PhD, gotten a lot easier to collect genetic data, even for non-model organisms. This is my stock next generation sequence slide. With this kind of data, we have access to analyses in a class of analyses we think of as population genomics, where we sample genomes across a landscape, and we can actually scan for associations within those genomes between genotype and the environments at the points of collection. So I want to say first off that there are, again, two different kinds of ways to go about this. There's methods where you go out and you collect many samples at individual sites, and then you perform tests based on differences in allele frequencies at those different sites. There's a huge wealth of methods associated with those kinds of sampling schemes. What I'm actually interested in is a much sparser sampling scheme, which is actually pretty common, where you have one individual representing each site that you collect at. So the individual variation is in geography. And the methods that we use to search for environmental associations in this kind of data are basically genotype environment association methods. There are sort of four broad categories, ways you can go about this. You can use methods that fit the environment as a function of genotype at your single nucleotide polymorphism, say. These could use a simple linear model. You can take that basic approach and say, well, I'm concerned about the confounding effects of population structure. So you can use methods that incorporate kinship in a mixed linear model. One of the most recent versions of this is incorporated into GAPIT, which is actually used typically in sort of non-spatial genome-wide association methods, but it can still be applied in these geographic studies. Then there's methods that flip this implied causal relationship in the regression model and fit the SNP genotype as a function of the environment in a logistic regression model. This was described by Just et al. In a 2007 molecular ecology paper. And there's a variant on that approach based on what are called generalized estimating equations that can again account for confounding variation across populations, and this is GEE. So these are the four methods I'm gonna take a look at, and I'll be using these 2- and 3-letter codes to indicate them throughout the talk. And I've actually done something like this already in a paper that just came out a couple months ago in Genetics. I took a sample of 200 lines of the model legube Medicago truncatula, and each of these dots on the map represents the location of an individual plant. And I scanned about 2 million SNPs diagnosed in all of those lines for associations with climate variation at the sites of collection. So this is association strength with annual mean temperature. Higher points have stronger, more significant associations. And this came up with a list of candidate genes that made good sense. But when I dug into the data, I also found that SNPs that showed strong climactic associations also lay in regions of the genome that differed in basic population genetic ways. So candidate SNPs, represented by the orange bars, lay in regions with increased haplotype frequency distortion. The most common haplotypes were more common, which is a sign of recent selective sweeps, compared to the gray bars, which are the genomic background. I also saw elevated FST between alternate alleles at those sites. And so that looked to me like we were beginning to get an idea of how selection had actually created those associations that I found in the scan. But you know, I actually wasn't sure. This was a little bit speculative in the discussion. Is this something that arises just as a consequence of the association being there, or is this reflective of how the association comes to be? And so the questions that I was left with after that study were, you know, how do we go about correcting for the confounding effects that we know are out there in natural populations, isolation by distance? How do we go about discerning which are the sites that we actually want to look at? Do we just use a strict p-value cutoff with a von Browning correction, or do we want to just take the tail of the whole genome-wide distribution of association values? And how do these association methods compare with sort of traditional population genetics, and how can they inform each other to tell us about how selection and local adaptation are occurring? So I simulated a whole bunch of data. I used the framework implemented in QuantiNemo, which is super user-friendly, and I highly recommend it if you want to simulate lots of population genetic data. I simulated a 10 by 10 array of populations with 100 individuals at each site, and 5% dispersal from site to site in a grid model like this. And I gave all of the individuals a single quantitative trait, which was under selection, and the optimum value for that trait varied with space. So depending on which row of populations you were in, the optimum trait value varied, but the strength of selection to be at that optimum was the same. And I set this up as either a clinal variation in the optimum or a sort of hard local adaptation scenario. The results that I have today are qualitatively the same for both scenarios, so I'll just be showing you clinal results. You can pretend that the talk is twice as long as it actually is. And then underlying this quantitative trait, I simulated either 1, 2, or 4 quantitative loci. These are going to be orange throughout the rest of the talk. And I put them on chromosomes with a bunch of linked loci, which will be in blue when their results show up. This let me ask about how changes at the quantitative locus affect variation and diversity at the linked loci. And I set up the adaptation to occur as a soft sweep. So the selected loci started with allele frequencies averaged across the whole population that reflected the frequencies that we would expect from a standard neutral model. So they were drawn at random at the beginning of the simulation from that distribution. And then I sampled one individual per population, and I tested for an association of environment, or actually the row number, and genotype in that sample. And I did also play around with different sampling schemes, drawing just 50 random individuals from the north half of the array and 50 from the southern half. I again got similar results to what you'll see. And so just looking at the simulation progress, here's FST between the north and south halves of the grid at neutral loci and selected loci. You can see that things are already looking like they've leveled out quite a bit by generation 1000. So I'm going to show you results from generation 2000. Okay, so I have those, remember I have those 4 different methods of testing for association. How did they stack up with these simulations? So here's the proportion of simulated loci that showed up as significant with an association p-value of 0.05 or smaller. For neutral loci, those are from a run where I just turned selection off, but it was otherwise the same. With 1 selected locus, with 2, or with 4. And you can see that all of these association models are very good at finding cases where there's just one selected locus, which is what we expect. And power to detect selective loci drops off quite a bit as we add more loci. Again, this is about what we're expecting. There's more variation in the number of false positives, neutral loci that look like they're significantly associated. The simple linear model is pretty bad in terms of false positives, but it's also better in terms of true positives compared to the MLM, the mixed linear model, which again has that control for population structure. That turns out to be just sort of universally conservative. It's bad at finding the real loci, but it's also good at avoiding the false ones. And the linear regression and the GEEs are actually logistic regression and the GEEs are actually more similar than I would expect. The controls provided in the GEE framework, at least for this case, don't seem to be avoiding false positives. If instead of a hard p-value cutoff, I use a relative p-value cutoff, if I just ask whether loci would end up in the top 5% tail of the genome based on the neutral expectation from the gray run, we get a pretty similar pattern, except that because I'm restricting the false positive rate based on the neutral distribution, I definitely have a false positive rate of 5%. So in a sense, if this tells you something you may have heard before. If you have lots of loci and you just take the tail, you know that your false positive rate is no bigger than the size of that tail. So you could use a simpler, possibly computationally faster method in that scenario. Linked loci, these are all for loci that are in complete linkage, actually at the same place on the virtual chromosome as the selected locus. Generally do not show the same strength of association as the actual selected locus. I think that's interesting because in these sort of genomic scan scenarios, we often think of ourselves as looking for loci that are in linkage with the true causal locus. This maybe should make you worry a little bit about power to detect things like that. So then information from population genetics. It turns out that at least FST differentiation between the north and the south tells us a lot of the same things we already know from linear regression-based association scan. But it does behave a little bit differently. So across all the simulated loci, there is a positive and just about one-to-one relationship between the strength of association, this is from the mixed linear model version, and FST, north-south, at the locus. Neutral loci in gray do show consistently lower association and lower FST. Linked loci sort of spread out all over the place. For just a one-locus model, we get higher association strength before we get separation in the distribution of FST. So the association is picking up on things that the FST distribution wouldn't. Once we add more selective loci, that sort of changes. We have everything definitely as a group has higher FST and higher association, but there's a lot of loci that we wouldn't discern as different from the neutral expectation. The interesting thing about FST, or another interesting thing about FST, is that it's kind of sensitive to starting conditions. So remember, I started selected loci with initial allele frequencies based on the standard neutral model. Selected loci in the one locus model that started with the highest allele frequency tended to have higher FST than those that started with lower initial frequencies. I think what's going on there is that those loci are just reaching FST equilibrium faster, but I'm not 100% sure about that. We don't find this issue with the association strength. Again, here's MLM results, and you can see that we're at about the same range of association strength regardless of starting allele frequency. So my results so far, and this is very, very tentative and initial, is again, no surprise, more loci, less power to detect them. The mixed linear model framework implemented in GAPIT with the kinship matrix control, does have the fewest false positives. Oh, but also the fewest true positives. And you can see all the rest there. If you have questions, catch up with me after. Thanks.